Hello and welcome. In this video, I'll introduce CryoREAD, a cutting-edge tool for de novo DNA-RNA structure modeling from Cryo-EM maps with resolutions up to 10 angstroms. So, CryoREAD automatically models nucleic acid structures through a series of steps. First, in the Cryo-EM map, it detects structural elements using deep learning, identifying locations of phosphates, sugars, and bases, and it also identifies 4 base types. Next, it clusters these detected positions to identify representative nodes, and then a backbone is traced using a graph constructed from these nodes. Then after that, CryoREAD assigns sequences to local fragments along this backbone path and then assembles them. Finally, it constructs full nucleotides according to sugar-phosphate base triangles followed by atomic structure refinement. And then you have your completed structure model. To use CryoREAD on the EM server, first navigate to em.kiharalab.org, scroll down to the roadmap section, and select CryoREAD. Now you'll need to prepare your input files. You'll need your Cryo-EM map in .mrc or .map format with a resolution between 2 and 10 angstroms, and optionally a FASTA file containing your nucleic acid sequence. If the FASTA file is not provided, CryoREAD will use the most probable nucleotide at each base position to construct the model. Here, upload your Cryo-EM map. In this example, I will model the microRNA in EMD 21051, which is at 3.7 angstroms in resolution. After uploading the Cryo-EM map, you will set your contour level. To do this, I'll use the Visualize with Set Contour Level button. I am going to pick a value such that I can clearly see my structure. .6 seems pretty appropriate for this map. Next, I will input my resolution, which is 3.7 angstroms in this case. Then I'll upload my FASTA file with my sequence. One thing to note is that if your structure has multiple identical chains, you want to copy the sequence in the FASTA file the number of times it appears in the structure. So suppose this sequence appeared 3 times in my structure, I will just copy this 3 times and name them something different. Okay, once everything is uploaded, all you have to do is click the upload button. Okay, this is the job results page. Currently, we can see that the job has been scheduled. If you don't have an account, you should bookmark this page or save the job ID so you can come back to this later. But if you're signed in, you can just go to Job Manager, My Jobs, and you'll be able to see this in the Job Manager. Once CryoREAD is finished, you can come back to this page and you will see your completed structure. The main output file is a PDB containing the modeled nucleic acid structure. You can also click the Show Map button to show the structure superimposed on the map. And to examine your results further, you can also download the PDB file and visualize using Pymol or Chimera or whatever you want by clicking this button. So in conclusion, CryoREAD is a powerful tool for automatically modeling DNA and RNA structures from Cryo-EM maps, and it's particularly useful for maps containing protein nucleic acid complexes. So, thank you for watching. For more detailed information, please check out our tutorial page or please read our paper published in Nature Methods. Thank you.